ನಮ್ಮ ಅಮೋಘಮ್ ಚಾನಲ್ಗೆ ಇವತ್ತು ನಮ್ಮ ವೀಕ್ಷಕರಿಗೆ ವಿಜ್ಞಾನ ಸಂಬಂಧಿತ ಕಾರ್ಯಕ್ರಮದ ಒಂದು ಝಲಕ್ ತೋರ್ಸಕ್ಕೆ ನಾವು ಬಂದಿದ್ದೀವಿ. ಎಲ್ಲರೂ ನೋಡೋಣ ಏನು ಮಾತಾಡ್ತಾರೆ. ಧನ್ಯವಾದಗಳು ಸರ್.
Now we get beautiful people like this man here who is standing with me. ಅವರು ಶ್ರೀನಿ ಕಾವೇರಿ ಅಂತ. ಹಿ ಈಸ್ ಎಮಿರಿಟಸ್ ಪ್ರೊಫೆಸರ್ ಪ್ಲಸ್ ಫ್ರಾನ್ಸಿಂದ ಬಂದಿದ್ದಾರೆ. ಅವ್ರ ಜೊತೆ ನನ್ನ ಡೀಲ್ ಇದೆ. ನೀವು ಯಾವಾಗ ಭಾರತ ದೇಶಕ್ಕೆ ಬರ್ತೀರಾ ಈ ಹರಟೆ ಅಂತ ನಡೆಸ್ತೀವಿ ನಾವು. ಸೊ ವಿಜ್ಞಾನ ಹರಟೆ ಅಂತ ಕಾರ್ಯಕ್ರಮ. ದಿಸ್ ಈಸ್ ನಾಟ್ ಅ ಲೆಕ್ಚರ್. ನಾವೇನು ಹೇಳ್ತೀವಿ, ಲೆಕ್ಚರ್ ಕೊಡಬೇಡಿ, ಏನು ದ ಸ್ಟೂಡೆಂಟ್ಸ್ ಆರ್ ಸಿಕ್ ಆಫ್ ಕ್ಲಾಸ್ ರೂಮ್ಸ್ ಆ್ಯಂಡ್ ಆಲ್ ದ್ಯಾಟ್ ಕೈಂಡ್ ಆಫ್ ಅಟ್ಮಾಸ್ಫಿಯರ್. ಸೊ ವಿ ಸೇ ಹರಟೆ ಮಾಡಿ, ಕನ್ನಡದಲ್ಲಿ ವಿಜ್ಞಾನ ಹರಟೆ. ಸೊ ವಿಜ್ಞಾನ ಬಗ್ಗೆನೇ ಇರ್ತದೆ. ಸೊ ವಿ ಡೂ ಔಟ್ರೀಚ್ ಪ್ರೋಗ್ರಾಮ್ಸ್ ಇನ್ ಸೈನ್ಸ್ ಆ್ಯಂಡ್ ವಿ ಗೆಟ್ ಬ್ಯೂಟಿಫುಲ್ ಪೀಪಲ್, ಎಮಿನೆಂಟ್ ಪೀಪಲ್ ಫ್ರಮ್ ಆಲ್ ಅರೌಂಡ್ ದ ವರ್ಲ್ಡ್. ಮತ್ತು ಇಲ್ಲಿ ಲೋಕಲಲ್ಲೂ ವಿ ಹ್ಯಾವ್ ಅ ಲಾಟ್ ಆಫ್ ವೆರಿ ನೈಸ್, ವೆರಿ ನಾಲೆಜಬಲ್ ಪೀಪಲ್ ಹೂ ಡೂ ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ವೇ ಅಂತ ಅದು ಇರಬೇಕು. ದಟ್ ಈಸ್ ದ ಫಸ್ಟ್ ಕ್ವಾಲಿಫಿಕೇಷನ್. ವಾಟ್ ಎವರ್ ದೇ ಟಾಕ್ ಅಬೌಟ್ ಅದು ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಇರಬೇಕು. ಇಟ್ ಹ್ಯಾಸ್ ಟು ಎಂಗೇಜ್ ದ ಯಂಗ್ಸ್ಟರ್ಸ್. ಅದೇ. ಸೊ ವಿತ್ ಅಸ್ ವಿ ಹ್ಯಾವ್ ಡಾಕ್ಟರ್ ಶ್ರೀನಿ ಕಾವೇರಿ. ಇವರು ಮಲೇಶ್ವರಂ ಹುಡುಗ. ಓಕೆ, ಹಿ ಈಸ್ ಅ ಪ್ಯಾರಿಸಿಯನ್. ಹಿ ಈಸ್ ಅ ಫ್ರೆಂಚ್ ಮ್ಯಾನ್ ಫಾರ್ ಮೋರ್ ದೆನ್ 45 years. ಬಟ್ ಜಸ್ಟ್ ಐ ವಿಲ್ ಗಿವ್ ಇಟ್ ಟು ಹಿಮ್. ಯು ವಿಲ್ ನೋ ಹಿ ಹ್ಯಾಸ್ ಹಿಸ್ ಹಾರ್ಟ್ ಇನ್ ಹಿ ಹ್ಯಾಸ್ ಅ ಕನ್ನಡಿಗ ಹಾರ್ಟ್ ಅಂತ. ಇವತ್ತು ಏನು ಮಾಡ್ತೀರಂತ.
ತುಂಬ ಧನ್ಯವಾದ. ಥ್ಯಾಂಕ್ಸ್ ಅಪ್ಪ. ಮತ್ತು ನನ್ನ ಹೆಸರು ಶ್ರೀನಿವಾಸ್ ಕಾವೇರಿ. ಇವ್ರು ಹೇಳ್ದಂಗೆ ನಾನು ಸುಮಾರು 45 ವರ್ಷದಿಂದ ಫ್ರಾನ್ಸ್ಗೆ ಹೋದೆ. ನಾನು ಅದಕ್ಕಿಂತ ಮುಂಚೆ ಪಶು ವೈದ್ಯಶಾಲೆ ಹೆಬ್ಬಾಳೊಳಗಿದೆ. ಅಲ್ಲಿ ನಾನು BBSc ಮಾಡಿ MBSc ಮಾಡಿ ಆಗ ಹುಡುಕ್ತಾ ಇದ್ದೆ. ನನಗೆ 3ನೇ ವರ್ಷ BBSc 3ನೇ ವರ್ಷ ಇದ್ದಾಗಲೇ ಮೊಟ್ಟ ಮೊದಲು ನಮ್ಮ ಇದು ಪ್ರಾಧ್ಯಾಪಕರೆಲ್ಲ ಇದು ಇಮ್ಯುನಾಲಜಿ ಕ್ಲಾಸ್ ಶುರು ಮಾಡಿದಾಗ ಇದು ಬಹಳ ಪ್ಯಾಷನೇಟ್ ಅನ್ನಿಸ್ತು ನನಗೆ ಆ ಸಬ್ಜೆಕ್ಟ್ ಬಗ್ಗೆ. ಅದಕ್ಕೆ ನಾನು ಅದರೊಳಗೆ ಹೇಗಾದರೂ ಮಾಡಿ ಸ್ವಲ್ಪ ಉನ್ನತ ಅಭ್ಯಾಸ ಮಾಡಬೇಕು ಅಥವಾ ಹೈಯರ್ ಸ್ಟಡೀಸ್ಗೆ ಹೋಗಬೇಕು ಅಂತ. ಆಗ ಇಮ್ಯುನಾಲಜಿ ಅಂತಂದರೆ ಯಾವ ದೇಶ ನೆನಪು ಬರಬೇಕು? ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್, ಇಮ್ಯುನಾಲಜಿ, ವ್ಯಾಕ್ಸಿನ್, ಅದು ಮೊದಲನೇ ದೇಶ ನೆನಪು ಬರಬೇಕು ಅಂತಂದರೆ ಲೂಯಿ ಪಾಸ್ಟರ್. ಸೊ ಅಕಸ್ಮಾತ್ ಪಾಸ್ಟರ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಒಳಗೆ ಅಕಸ್ಮಾತ್ PhD ಮಾಡೋಕ್ಕೆ ಅವಕಾಶ ಸಿಕ್ಕರೆ ಎಷ್ಟು ಚೆನ್ನಾಗಿರುತ್ತಲ್ವ ಅಂತ ನನ್ನ ಭಾವಿಕೆ. ಆದರೆ ನನ್ನ ಅದೃಷ್ಟ ಅದೇ ವೇಳೆಯಲ್ಲಿ ಫ್ರೆಂಚ್ ಗೌರ್ಮೆಂಟ್ನವರು ಇದು ಫ್ರಾನ್ಸ್ವಾ ಮಿತ್ತೆರಾನ್ ಅಂತ ಒಬ್ಬರು ಆ ಪ್ರೆಸಿಡೆಂಟ್, ಆಗ ಸೋಷಲಿಸ್ಟ್ ಟೈಪು, ಆಗ ಅವರು ಬಹಳ ಓಪನ್ ಅಪ್ ಮಾಡಿದ್ರು ಈ ನಮ್ಮಂಥ ಹುಡುಗರಿಗೆ, ಬೇರೆ ಬೇರೆ ದೇಶದಿಂದ PhD ಮಾಡೋರಿಗೆ, ಹೈಯರ್ ಎಜುಕೇಷನ್ಗೆ. ಆಗ ನನಗೊಂದು ಒಳ್ಳೆಯ ಅವಕಾಶ ಸಿಕ್ತು. ಅಲ್ಲಿ PhD ಮಾಡಿದ ತಕ್ಷಣ ನಾನು ಇದು ಪ್ಯಾಶ್ಚರ್ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಮಾಡಿದ್ರೆ ನನಗೆ ಆ್ಯಂಟಿಬಾಡೀಸ್ ಬಗ್ಗೆ ಆವಾಗಲೂ ಇಲ್ಲಿ ಇಂಡಿಯಾ ಒಳಗೇ ನಾನು ಭಾರತದೊಳಗೆ ಪಶುವೈದ್ಯ ಕಾಲೇಜೊಳಗೆ ಸ್ವಲ್ಪ ರಿಸರ್ಚ್ ಮಾಡಿದ್ದೆ. ಯಾಕೆಂದರೆ MUS ಒಳಗೆ ರಿಸರ್ಚ್ ಇರ್ತಿತ್ತು. ನಮ್ಮ ಕಾಲೇಜ್ ಬಹಳ ಚೆನ್ನಾಗಿದೆ. ನಮ್ಮ ವೆಟನರಿ ಕಾಲೇಜ್ ಬೆಂಗಳೂರು ಪಶುವೈದ್ಯಕೀಯ ಕಾಲೇಜು, ಅಲ್ಲಿ ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ಮೈಕ್ರೋಬಯಾಲಜಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್, ಇಮ್ಯುನಾಲಜಿ ಬಹಳ ಚೆನ್ನಾಗಿ ಆಗಲೇ ಬಹಳ ಸ್ಕೀಮ್ಸ್ ಇರ್ತಿತ್ತು, ಇನ್ಫ್ರಾಸ್ಟ್ರಕ್ಚರ್ ಎಲ್ಲ ಚೆನ್ನಾಗಿರ್ತಿತ್ತು. ಸೊ ಅಲ್ಲಿಂದ ನನಗೆ ಅವಕಾಶ ಸಿಕ್ತು. ಆಮೇಲೆ ನಾನು ಆ್ಯಂಟಿಬಾಡೀಸ್ ಬಗ್ಗೆ ನಾನು ಇಡೀ ಜೀವನ ಎಲ್ಲ ನನಗೆ ಗೊತ್ತಿರೋದು ಒಂದೇ ಒಂದು ವಿಷಯ, ಆ್ಯಂಟಿಬಾಡೀಸ್. ಇವತ್ತು ಕೇಳ್ತಿರಂತೆ, ಬೇರೆ ಏನೂ ಗೊತ್ತಿಲ್ಲ ನನಗೆ. ಸೊ ಅಲ್ಲಿಂದ ನಾನು ಅಮೇರಿಕಾಗೆ ಒಂದು 3 ವರ್ಷ ಇದ್ದೆ. ಅಲ್ಲೂ ಕೂಡ ಥೆರಪ್ಯೂಟಿಕ್ ಆ್ಯಂಟಿಬಾಡೀಸ್ ಬಗ್ಗೆ ಒಂದು ರಿಟುಕ್ಸಿಮ್ಯಾಬ್ ಅಂತ ಒಂದು ಬಹಳ ಪ್ರಖ್ಯಾತ ಅಂದರೆ ಏನು, ಇಟ್ ಈಸ್ ಎ $multi-billion ಬಿಸ್ನೆಸ್. $multi-billion ಬಿಸ್ನೆಸ್. ಆ ಆ್ಯಂಟಿಬಾಡಿ, ಮೊನೊಕ್ಲೋನಲ್ ಆ್ಯಂಟಿಬಾಡಿ ವಾಸ್ ಬಾರ್ನ್ ಇನ್ ಫ್ರಂಟ್ ಆಫ್ ಅಸ್. ಓಕೆ, ಅಂದರೆ ನಾವು ಯಾವ ಪೋಸ್ಟ್ ಡಾಕ್ ಎಲ್ಲಿ ಯಾವ ಲ್ಯಾಬ್ ಒಳಗೆ ಮಾಡಿದ್ನೋ ಅದನ್ನು ಅವರು ಮೊಟ್ಟ ಮೊದಲು ತಯಾರಿಸಿದರು. ಈಗ ಜಗತ್ತಿನೊಳಗೆ ಈಗ ಹೋಗಿ ಇಲ್ಲಿ ಪಕ್ಕದೊಳಗೆ ಹೋಗಿ ಅದನ್ನು ಉಪಯೋಗಿಸ್ತಿರ್ತಾರೆ. ಅದನ್ನು ನಾವು ಸ್ಥಾಪಿಸಿದ್ದು, ನಾವು ಎಸ್ಟಾಬ್ಲಿಷ್ ಮಾಡಿದ್ದು. ಎನಿವೆ, ಸೊ ನಾನು ಅದ್ರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ಮಾತಾಡ್ತೀನಿ ಇವತ್ತು. ಮತ್ತು ಇನ್ನೊಂದು ನ್ಯಾಚುರಲ್ ಆ್ಯಂಟಿಬಾಡೀಸ್ ಬಗ್ಗೆ ಮಾತಾಡ್ತೀನಿ. ಅದನ್ನು ನೀವೇ ಕೇಳ್ತೀರಂತೆ ಒಂದು 5 ನಿಮಿಷ ಆದಮೇಲೆ. ಓಕೆ, ಥ್ಯಾಂಕ್ಸ್.
Good evening, everybody. Good evening. Again, good evening, everybody. Somebody said good morning. I'm so happy to hear that. There is at least one Homo sapien being here. Again, good evening, everybody. Thank you so much. This is my test to see how many Homo sapiens are there in the room and how many are Homo erectus. So Homo erectus are people who know how to stand erect. Yeah? And Homo sapien is, you all have come from biology background, right? You know Homo sapiens? Yeah, we like saying it twice. It's such a nice word. Yeah? We want to be a land of Sujalam Subalam. We want a beautiful world. Do we want a beautiful world? Anyone? Do we want a beautiful world? Is the world we are living in right now beautiful? Is it beautiful? Is it beautiful? The world is beautiful. The things that are happening that we humans, so-called Homo sapiens, we are doing to this world, is it nice? Yeah. The only way we can make this world beautiful is by us becoming beautiful first. Remember, if you have something, only then you can give it. If you have anger, you will only share anger. If you have depression, you will only share your anxiety and your depression. If we are happy, beautiful people, that is what we will share with this world and make it beautiful. We have to be beautiful first. I'm going to, I have brought this beautiful, beautiful person with me. I've been living in France for more than 45 years, but he's also a Bangalore boy. Malaysian Buddha. He will talk to you in perfect Kannada. The next moment he will talk to you in perfect French. The next moment he will talk to you in whatever language you are familiar with. He's a polyglot, a polymath. He's in Bangalore he says Jajabar or wanderer. Except for North Pole and South Pole, he has been all over the world. Yeah? That's why this man is beautiful, because he's a Vishwamana. All these people are Vishwamanas. And I had the good fortune of working with a lot of these kind of Vishwamanas. Yeah? Proud to be a Tridigo, proud to be an Indian, that's nonsense. The whole world is yours.
To be back in Mysore. I cannot detach Mysore from myself. It is a wonderful place. And thanks to Shubhankar and Sitaram and the very organization, and to you, sir, Hanumantacha Joshi, that I am here, and I am extremely happy to share some of our thoughts, some of our ideas, some of our results, some of our research findings. And more importantly, I would like to see how we as a research people can interact with you youngsters and stimulate or provoke some of the interest in research in you people. And at the end, I'll also provide some tips as to how, particularly the young people at the back, you can look for higher education opportunities in the European Union and particularly in France. And this also includes the scientists, young faculty people who are here to establish collaboration with the European Union and France. Looking forward for an exciting discussion with you all. It will not be a one-way traffic. It will be a herte, almost pele herte. Okay? Thank you very much.
Thank you, sir. We're extremely grateful to have Dr.
Respected Dr.srinivas.v.kaveri, Emeritus Director of Research, CNRS, Sorbonne University, Paris, Director of CIUP, Paris. The guest of honor of today's function, Shri Shubhankar Biswas, Executive Secretary, tAcT. Respected Sitaramu sir, and all the distinguished professors and very young people who are sitting in the front. First of all, thank you for coming to the institution, and a hearty welcome to you once again, sir. We had a small interaction before starting of this program, and Srini sir was, such a simple and wonderful human. In 15-20 minutes we could understand better and what exactly they are doing, for the sake of the profession of health and science. Thanks to Sitaramu sir, very recently we had met when Mr. Narayana Murthy was here for a program. We had a small talk. Sitaramu sir came with Mr. Sundaramurthy, and he said that we would like to conduct some programs, and it was a boon in disguise. ತುಂಬ ಖುಷಿ ಆಯಿತು. ಯಾಕಂದರೆ ಇಲ್ಲಿ ಸುಮಾರು ಜನ ಸೈಂಟಿಸ್ಟ್ ಬಂದೋಗಿದ್ದಾರೆ, ಬಟ್ ಅನಾಯಾಸವಾಗಿ ಅವರೇ ಬರುವಂಥದ್ದು ವಂಡರ್ಫುಲ್ ಅವ್ರು ಹೇಳಿ ವಿಶ್ವಮಾನವ ಅಂತ ಹೇಳಿದರು. ತುಂಬ ಖುಷಿನೇ ಆಯಿತು. ನಂತರ ಇನ್ನೊಂದು ಗೊತ್ತಾಗಿದ್ದೇನೆಂದರೆ ಅವರು ಈಸ್ ಫ್ರಮ್ ಬಾಗಲಕೋಟೆ ಬೇಸಿಕಲಿ, ಸ್ಪೆಂಟ್ ಆಲ್ಮೋಸ್ಟ್ more than 45 years in France, ಬಟ್ ಬ್ಯೂಟಿಫುಲ್ ಕನ್ನಡ, ಅದರಲ್ಲಿ ಯಾವುದೇ ರೀತಿಯ ಬೇರೆ ಟಚ್ ಇಲ್ಲ, ಏನಿಲ್ಲ. ವಂಡರ್ಫುಲ್ ಪರ್ಸನ್. ಇವ್ರು ನಿಮಗೆ ಗೊತ್ತಾಗುತ್ತೆ ಮಾತಾಡ್ತಾ. ಸರ್, ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ಥ್ಯಾಂಕ್ಸ್ ಯು ಫಾರ್ ಸ್ಪೆಂಡಿಂಗ್ ಯುವರ್ ವ್ಯಾಲ್ಯೂಬಲ್ ಟೈಮ್ ಹಿಯರ್, ಆ್ಯಂಡ್ he will be here for a very brief period of time. And without taking much time, I thank all the people who are here, and especially for my students. Please listen to his talk very carefully and try to understand. And if you have any questions, you can definitely ask him. And sir is here for not only delivering a talk, he is also here to guide you. ಫಾರ್ಮಸಿ ಅವರಿಗೆ ಏನಾದರೂ ಆಪರ್ಚುನಿಟಿ ಇದೆಯಾ ಫ್ರಾನ್ಸಲ್ಲಿ, ಅದರ ಬಗ್ಗೆನೂ ಸ್ವಲ್ಪ ಹೇಳ್ತಾರೆ. ಆ್ಯಂಡ್ he will also guide you, how to prepare yourself also along with his lecture also. So thanks to him. Thanks to Shubhakar Das ji also. We will be in regular touch, and we have already spoken a little during that. And there are some internship facilities available for B.Pharm that we will be talking once again about that. It is a wonderful day today. Thanks to Professor Kulkarni also for coordinating, and a lot of thanks to Sitaramu sir especially for bringing this one. And ನಿಮಗೆ ಏನೇ ಡೌಟ್ ಇದ್ದರೆ ಕೂಡ ಕೇಳ್ಕೊಳ್ಳಿ. If not exactly on the subject of antibodies, you can definitely talk to him what are the opportunities available, how do we prepare ourselves, in the field of research. So with this, thanks to all, and have a nice time. Thank you.
Wired that I feel I'm a robo. The robos, how they walk? Maybe I should be making. Anyways, it's okay. That sound is a little. Easy to correct.
Mics are there, no, it's disturbing.
One you can put on one mic ಬೇರೆ ಕಡೆ ಹಾಕಿ, ಇಲ್ಲಾಂದ್ರೆ ಪಾಕೆಟ್.
All of you at the back, maybe you don't want to hear me anyway. That's why you are sitting at the back, right? Yeah. But I'll come to you, don't worry. Okay, I'll come to you. Yeah. I won't be staying here because my entire talk, this is my first disclaimer. My entire talk is not for the same people here, people who have the same hairstyle as me, meaning more style and less hair. Or no hair. You see? Yeah. My talk is essentially for you people, okay? So I don't want you to sleep, please. All right? So this story is all about cricket. Nah, it's not. It's the title says a century. You know what a century is, right? And still batting strong. Okay? We are talking about some very interesting molecules here. I do not know whether you can see the slide there. Yeah? What do you see there? It's a group of people sitting, right? We are in the heart of Paris there. How many people have been to Paris? Not many. You will get a chance. Don't worry. You will get a chance. It's a beautiful city. Right in the heart of Paris, this place is called Jardin du Luxembourg. It's a French word meaning the Luxembourg garden. And my lab is behind that Senate building. That what you see is the Senate. It's right in the heart of Paris. If you walk on the right side of that, about 200 meters or so, you will be in a place called Panthéon. Panthéon is a place where all great minds, great lives, great artists, great literary people, some scientists, Voltaire, Victor Hugo, Guy de Maupassant, people like Pascal, must have heard of Pascal's law, Louis Braille, Braille, yeah. Those people, they're all buried there, okay? It's a very, very, very important place in Paris, and that's right in the heart of Paris. And behind that building that you see is my lab. There is a medical school, Faculty of Medicine. On the 4th floor is my lab. And these people work with me, and I don't know whether you see them closely. You cannot probably see their faces. We have people from all over the world. We have people from African continent, South America, Europeans, China, Korea, many places. And I have some affinity, I don't know why, for the Indian people. I don't know why actually. And these people, there are actually on the very right side, you have Vani. She is now associate professor in biotechnology in IIT Madras. And you don't see some people here. This is Mysore, not Madhur Mohan. He's from Madhur, not far from here, you know, okay? And so the people at the top, they're all from different parts of India, but they were mostly trained in the Indian Institute of Science, Bangalore. And for some reasons, I keep going to Bangalore. I'm from Bangalore. So whenever I go to Bangalore, I meet these young people. If they're interested in doing research, I said, why don't you come to my lab? And either they come for a PhD or a postdoc or, you know, things like that. Okay. And I'm thankful to them. And I'm also thankful to many clinicians who help me, because I am a veterinary doctor, but most of the questions that I ask have a clinical bearing, are very closely related to the clinics. And so I work with doctors who are listed on the left side, and people on the right side are international collaborations. I collaborate a lot with India, particularly in Jipmer, Pondicherry, KEM Hospital, Indians of Science, all over. I like to collaborate with many people, okay? And they all help me in many, many ways. So today, for people who are not in biology, it may be a little bit off. That is only for the first 2-3 minutes. If you are patient with me for, I know you are a chemical engineer. Chemical? Yeah? Yeah, you are an engineer. Yeah. So if the first 2 slides, if you can bear with me, then you will be fine. Okay, the rest is a story. I have a story to tell you because I like stories. Okay, so the story, the very first thing that starts is, as you are sitting here, whether you want it or not, you are confronted with pathogens, microbes. They're all around you, whether you want it or not. You are breathing them, okay? And not all of them are extremely dangerous. They can be, but they're not necessarily all dangerous. But they're coming and getting you. They want to infect you. They want to attack you. And it is at that time, there is what is called innate immune compartment. I'm sure you all learnt in your 7th standard or 8th standard what is innate immune system. Maybe you did not call it that way. You have cilia, you have hair, you have mucus, you have saliva, you have tears, and there are some cells also in the body. As the microbe goes, either through skin or through mouth or through eyes, whatever route, this innate immune system takes care of them. It eliminates very easily, and that's the role of innate immune system. However, if the pathogen, if the organism, the microorganism is very toxic, very virulent, and it comes in a big number, you remember what happened in coronavirus infection? If you had had a dinner with your girlfriend or boyfriend the previous evening, the next morning when you woke up, and if the boyfriend or girlfriend had the coronavirus infection, he or she does not know, but he or she transmitted to you in one morning, every cell that was infected made 1,000 copies of that virus. Are you with me? Just because you had a dinner with your girlfriend. No, it's not that. The virus enters, and within one night, within 16 hours, it could multiply to such an extent that every cell that was infected had 1,000 copies of that. These are very virulent, very strong, and at that time, it is your innate immune system may not be able to take care. Are you with me? So you need something more solid, something more specific, because innate is like cilia, tears, saliva. Those are not very specific. Whereas you need something very powerful, targeted, and that is the role of adaptive immune system. It's called adaptive because it adapts, specifically adapts to the pathogen and tries to attack and kill the pathogen. It's like when I was very young, I used to go from Malleshwaram. You heard that I'm a Malleshwaram boy. I used to go on a bicycle from Malleshwaram to Hebbal. Hebbal is where the veterinary college is, okay? And In between, there was this Sadashiv Nagar, red light, signal light. And sometimes the signal light I was probably not seeing properly, and I used to sneak through. And sometimes I used to get caught by Constable Ramaya. And Ramaya would say, "What is this, young man? What are you doing? Why are you skipping this red light? You should not do this. It can be dangerous." I said, "Oh, sir, I did not know it was red. I thought it was orange, maybe deep orange or dark orange, but it was red." But he would say, you negotiate a little bit with this Constable Ramaya, and then you manage to somehow, a little bit, you know what I mean, okay? Somehow you escape. But imagine I'm a very famous film star, and I'm drunk, I have had a few drinks, and I'm driving a big SUV car, and then I run over a few people who are sleeping on the footpath. You know what I mean? And that time, Constable Ramaya cannot help. You are taken to the closest police station. They ask your ID card, they ask your driver license, they see whether you are drunk or what other substances you had, and then accordingly you will be treated. You will be punished or other things. Similarly, if the pathogen is very, very virulent, they take it to the closest lymph nodes. There are lymph nodes all over your body, and they are called different lymph nodes. And in the lymph nodes, it's a pocket where you have first encounter with the immune system. The pathogens are now coming into the lymph nodes, and in the lymph nodes, there are some specialized cells. They are called antigen presenting cells. Don't worry too much about it. You can forget the name. But the point is, they take up the pathogen, they cut into pieces and show it to the immune system, to the T lymphocytes, says, "This is a pathogen, we want to do something about it." And the T cells get activated. They start multiplying. They want to kill this pathogen at any cost. They multiply and they start secreting so many molecules, and some of these molecules, they give the signal to the B lymphocytes. T, B, don't worry, there are different, we'll worry about. Those people will worry about, you don't have to. The B lymphocytes, they were sleeping, now they wake up, they start making antibodies. B lymphocytes make antibodies. Are you with me? And the B lymphocytes make antibodies, and antibodies, unlike the innate immune system, these antibodies are very specific. And imagine by that time, the pathogen has already reached the target organ being lungs. Let's imagine. If you have a question, you can stop me. Anybody, if you have any questions, please stop me in the between, okay? No hesitation. Okay, it's a herte, you know that. So, the pathogen has reached the target organ, which is lungs, for example. And there, the antibodies that are very specific, they can recognize the pathogen. Moment the antibodies shown here as Y-shaped molecule, the antibodies are always shown as Y-shaped molecules like that. And through the arms, they interact with the antigen, which is the pathogen, which is already in the lungs. And through the legs, they interact with another molecule called Fc receptors. You must have read about Fc receptors. And one moment you engage Fc receptors on the macrophages, inflammation begins. And this inflammation is very good. When you have an infection, you have inflammation. Inflammation is associated with some fever, some pain, some swelling, and many such symptoms. But don't worry, this is natural. Immune system is trying to fight. It is trying to get rid of the pathogen, and therefore you have to pay a small price. Okay? So the pathogen is trying to be eliminated. But if that is not sufficient, then other molecules secreted by the neutrophils, they come into picture. They also attack. We have to get rid of the pathogen at any cost. And even if that is not sufficient, there is one more molecule in the immune system, immune system, and that's called complement. And that also will start along with the fight. You have a whole army, one after the other. The whole objective is we have to kill the pathogen, get rid of the pathogen. They all work in tandem, and the complement also tries its best. Now, even if that is not sufficient, the T lymphocytes, they also wake up. The T lymphocytes, oh, this pathogen is not ordinary. This guy needs more strong immune response. So the T lymphocytes, they start secreting other substances. Ultimately, they eliminate the pathogen. They differentiate, they do something, but they eliminate the pathogen. And that's what happens in your body practically almost any other day. Okay, but you don't even realize that. You don't even realize. Only when the infection is very heavy and your body is reacting very strongly, in that case you feel the fever, the pain and all that. It is then that you feel, oh, there is something going on. Okay, otherwise immune system is working all the time. Okay, now there's one advantage of this. There is a bonus. Some cells go as memory cells. Some T cells, some B cells, they sit in the corner. They say, oh, I know this pathogen. I know this bacteria. If you come next time, I'll take care of you quickly. And that is the concept behind vaccination. When vaccination happens, you have a little fever the first day. You must have noticed that. Little uneasiness. But that is because it prepares the body for these so-called memory cells. The memory cells, they remember the pathogen. And the pathogen, when it comes next time, that pathogen is eliminated very quickly, and that's the advantage of vaccination. Now, there's something which is very unusual. This immune system, I told you, it's very strong, right? It's very strong. It's very, very potent. But if there is any of these components is missing, any of them is missing, then you will be immunodeficient, meaning you will not be able to fight the pathogen normally. There are children, unfortunately, that are born without the immune system, or at least one of the components. And these children cannot fight the infection. They die very young, because for even a smallest infection, they cannot cope up. They have to. Only in the last few years, we have started identifying. I'll come to that. But you should understand that there are some children that can be born without one of these components, and these are called immunodeficient. And these are genetic inborn diseases. It's not acquired from outside. It's not because of an infection. It is their parents. One of those parents they give. It is the X-linked disease. And you may be missing one of these. You remember that big poster there? In that big poster, if anything is missing, then you are immunodeficient. You will not be able to fight the infection. You will suffer. And this genetic disease has to be treated a different way. And the way, one of the most modern ways of treating is through immunotherapy. And this person there, you see Alan Fisher, he is the one who first time tried how to correct the genes at the very bottom. If it is a small infection, you give antibiotics. But if it is genetic, you need to correct the gene, right? The gene that is responsible for this defect. Hemophilia. You heard of hemophilia? You have heard of hemophilia? Yeah? Have you heard of hemophilia? Yes?
Yes, sir.
What is it? Don't worry if it's a wrong answer. Say something. Hemophilia. What happens there? Don't remember? Who remembers hemophilia? You do? Yeah. The blood does not coagulate. Even if you have a small toothbrush, problem, the boys mostly, okay? Or when the boys are shaving, they can bleed, and they can bleed non-stop because they are missing one of the molecules necessary for coagulation. That's called factor 8. And that's a genetic disease. You can treat the disease by giving alternate substance to fill up the gap. But the best way is to treat at the very bottom, right? That is genetic. You can do the gene therapy, and that is the basis of gene therapy. Try to understand what gene has gone wrong, where it has gone wrong, and that's called gene therapy. Now, these are very sophisticated treatments. Now, we'll talk about that later. For the next 3 or 4 hours that you are with me, no, I'm kidding. You are already started worrying about your lunch and no. I'll leave you in next 20-25 minutes, okay? I'm going to talk about one molecule, okay? That is antibodies. How many people have heard of the word antibodies? Antibody. You have heard the word antibody? Yes or no? Huh? No one knows antibody? Raise your hands. Those who have heard the word antibody, please raise your hand. You have not heard the word antibody? You have. Why did you not raise your hand? You have heard the word antibody? Yes? Okay. So today we are going to talk only about antibodies. Alright? So the story starts approximately 120 years ago when a German scientist along with a Japanese scientist, the German scientist his name is von Behring, and Adolf von Behring was doing very beautiful experiments along with a Japanese scientist called Kitasato. What they were doing was, by then, we are talking about 1890s, and in Germany, already Robert Koch had known that some pathogens can cause some disease. And in France, Louis Pasteur, you heard of Louis Pasteur? Louis Pasteur had happened. Louis Pasteur had demonstrated that there are pathogens, there are microbes, there are bacteria, there are viruses, there are fungus. These can cause some diseases, okay? And they had started identifying more and more pathogens. What the German did was he injected either a small quantity of this pathogen, okay, either inactivated or took the toxin and inactivated and injected into the animals, and took the serum from the animals and treated other animals that were infected with the toxin or with the bacteria. Are you with me? So he first prepares the anti-serum, meaning he first injects the pathogen in another animal and then takes that and injects into the animal which was sick, and he cures the animal, the second animal, of this disease. Meaning you can take the serum from the first animal, give it to the other animal, you can actually treat that. And that is when he knew there is something in that particular plasma or serum of the first animal which can cure the disease in the second sick animal. And it is at that time they started looking what is in this serum or plasma which can cure the disease. And that is the beginning of the discovery of antibodies. The first time they used the word antikörper, which is a German word. Soon it was translated into English as antibody, into French as anticorps, each one translated. And if I translate that into Kannada, that could be Pratikriya Kanagalu, something like that. Because Kannada we have not practiced so much for scientific language. Pratikriya Shastra is immunology. Pratikriya Krama is immune system or immune response. Pratikriya Kanagalu, ಈಗ ಬಿಳಿ ರಕ್ತ ಕಣಗಳು, ಕೆಂಪು ರಕ್ತ ಕಣಗಳು ಇದೆಲ್ಲ ಕೇಳಿದ್ರು. ಪ್ರತಿಕ್ರಿಯಾ ಕಣಗಳು ಅಂದರೆ ಅದನ್ನ ಆಂಟಿಬಾಡೀಸ್ ಅಂತ ಹೇಳ್ಬೋದು. ಎನಿವೇ ಫಸ್ಟ್ ಹೇಳಿಮ್ಮ, ಪ್ರತಿರೋಧಕ. ನಾನು ಹೇಳ್ತೀನಿ ಪ್ರತಿಕ್ರಿಯಾ ಅಂತಲ್ಲ, ಪ್ರತಿರೋಧಕ. ಐ ಮೈಂಟ್ ಪ್ರತಿರೋಧಕ, ಸಾರಿ, ಪ್ರತಿರೋಧಕ. ನೋ, ಪ್ರತಿರೋಧಕ ಇಸ್ ದ ರೈಟ್ ವರ್ಡ್. ಪ್ರತಿರೋಧಕ, ನಾಟ್ ಪ್ರತಿಕ್ರಿಯಾ. ನಾನು ಪ್ರತಿಕ್ರಿಯಾ ಅಂತಲ್ಲ, ಸಾರಿ, ಐ ಟೇಕ್ ಬ್ಯಾಕ್. Yeah. Kriya is reaction. That's immune response. But Pratirodhaka Shastra, sorry, I stand corrected. So, the moment the antibodies was discovered, people started looking at different aspects of this. What is the structure? What is the function? What can we do with this? How can we use these antibodies? Because by then, they already knew that we can treat some diseases. And it is at that time that these other doctors, they started purifying more and more antibodies from more and more animals, more and more diseases. So now they had antibodies against many pathogens. Earlier these pathogens were killing the children, killing the human beings. Now we have an antibody in the hand. Are you all with me? Before that, of course, there were no antibiotics, there were no drugs, there was no way of treating. But now we have antibodies in the hands, and we can start treating these. And this was a major discovery. This was very important. And many years passed, Many Nobel Prizes were won because these antibodies became very important subject. Many biochemical studies were done, structural studies were done, and fantastic discoveries made. Allergy, what is allergy because of antibodies? So many discoveries made, so many Nobel Prizes were won. But then came the bad news. The bad news came in the form of the World War II. You must have heard what happened in the World War II. Several million people died, several soldiers died. They were not dying only on the battlefront. They were not dying only on the war front. They were dying also in the hospitals because they had infections. No antibiotic yet, no penicillin yet. And the only way they could have been treated was with antibodies. So it is at that time that the President of the United States of America commissioned a certain Edwin Kahn in Boston to prepare gallons and gallons of antibodies. You can prepare from normal human beings. You don't even have to have specific. Give blood, donate blood. And there was a huge donation campaign, and thousands of people donated all over the country. They started collecting thousands and thousands of gallons of blood and purifying that. Because this man here, he knew how to purify different fractions. By then, by the time the antibodies were discovered, and now we knew more about the biochemistry, we knew how to purify. It was a good, beautiful, pure antibodies now. And these antibodies, they saved several thousand healthy people. Several thousand people suffering because of the infectious diseases. The soldiers, they were dying, so they improved them. And that time, this person here, Colonel Bruton, he came from the war too. And he had known that there are some diseases that are called primary. And one day he had this young boy who comes and he was treating with antibiotics because he was a pediatrician. He gave antibiotics. The boy is not recovering. The boy is not responding, and he tried whatever possible antibiotics. What is going on with this little boy? Why is he not answering? Why is he not recovering? And then he thought maybe there's something wrong with his immune system. And then he looks at on the top you see a normal, like you and me. What is your blood picture? You have different fractions in your blood plasma. And he compared that with the patient, with the little boy. And what he saw was that this boy did not have antibodies at all. The peak that corresponds, the protein here this peak corresponds to the antibodies. The boy was born without antibodies. Genetic disease. So for the first time, we knew that some kids can be born without antibodies. And then he said, why don't I treat with antibodies? Well, now we have antibodies because we know. Ailment was repeated infection. The boy was having cough, respiratory disease, and then the sneezing and pneumonitis, sinusitis. So these are normal infections. Normally you get over, when you have a common cold in 3-4 days with the drugs, or 72 hours without drugs. With drugs, without drugs, it's the same with the common cold, And some infections you can treat with antibiotics. You know that you take too many antibiotics these days, right? So this boy is not responding to any antibiotics because it's a genetic disease, and he needs this push from the antibodies. And it is at that time the doctor, Colonel Bruton, gives the antibodies to this boy. Within 4 or 5 days, the boy is jumping around. Within next day, actually. Then he understood that some kids can be born with a defect, but now we have a treatment. Treatment is, these kids were dying because repeated infection, you die, because how much more your body can take. And they said, okay, there is child-borne disease. They didn't know what was going on. Now we knew there is a genetic disease. We can treat with these antibodies. And we discovered many such diseases that were related. But the injection that was given was intramuscular, and it was a thick paste, actually, antibody paste. Some paste, it was not very convenient. This man here, he showed that you can actually treat these molecules. The paste becomes liquid, infusion, then you can actually treat intravenously. Imagine when you get an intramuscular dose, you become an abscess, and every 4 weeks you have to be treated because these boys don't have. So instead of giving muscular, now they started getting intravenous. So now onwards, for the next few minutes, you hear the word intravenous immunoglobulin. What are immunoglobulins? They are antibodies. Okay, so intravenous immunoglobulins came into picture for the first time in 1950s. And this man, he is a very famous immunologist. This is the father, Charlie Geneve, and this is the junior. Okay, and he has written several books, Charles Geneve. Those people who want to study immunology should look up Charlie Geneve's books. He's very good books. Anyway, so soon people started identifying we can treat these diseases. Many diseases we started treating, and they identified. And in the 80s, on one cold October, you are going fast forward, right? On one cold October, drizzly, depressive morning, cold October, depressive, drizzly, rainy, cloudy morning. Okay, I land in Paris. Nobody noticed that. But luckily, I met this person. You see the person? You are not looking at the right person. Okay? Yeah. No, you are not looking at the right person. I met this man here. Okay? But the other person was more attractive, right? Anyway, so I met that person, and with whom we started asking many questions. And it is at the same time something very strange happened in Europe, particularly in Switzerland. Until now, we knew that we can treat immunodeficiency, meaning children that do not have immune system correctly, they're deficient in immune system, you can treat them with antibodies. This particular doctor, Paul Imbach, gets another little boy who has a disease which is called autoimmune disease, where the boy has antibodies that are directed against his own platelets. And the platelets are very important cells in your body that help in coagulation. And what happened to this boy was the autoantibodies were binding to these platelets and the phagocytes were eating away. So which means you did not have any platelets. The platelets were going down. But the man, this young boy, also had hypogammaglobulinemia, meaning the antibody levels were very low. The doctor was puzzled. The boy has an autoimmune disease, plus the boy also has an immunodeficiency. What should I do now? He said, look, I know how to treat immunodeficiency by antibodies. Let me give antibodies, at least let me save him and see what happens. When he gave the antibodies, within 4 or 5 days, the platelet numbers came up to normal.
Antibody specific or is a common one?
Which
For all
one?
disease.
The one who gave or one who the boy had done?
General I'm asking.
Yeah.
Antibody.
Yeah, the antibodies that we infused into this boy, very good question, not specific at all. These antibodies from you healthy people, we pool the antibodies from several thousand healthy plasma donors, and you make a pool, super pool of antibodies, you purify, and we give it to the patients that do not have antibodies. But this boy has excess of autoantibodies. Autoantibody means an auto is self. Yeah, exactly. Autoantibodies against platelets. And within 4 days, the autoantibody levels came down and the boy was happy. So which means these immunoglobulins or antibodies we thought were useful only for immunodeficiency, we now started seeing they have something else. And suddenly we started getting interest. We treated autoimmune hemophilia. Now onwards I use the word autoimmune. You know what autoimmune means, right? The immune system, it goes wrong and is directed against the self. And it's called autoimmune. There are many types of autoimmune diseases. Now I think more than 150 different kinds of autoimmune conditions have been identified. So what happened is that we wanted to treat a immunodeficiency, but now we also got a treatment for autoimmune disease. But we didn't know how it worked. Okay, and then we started using it in many diseases, in all these diseases. I don't want to go into the details. Maybe you know them already. ITP, Kawasaki disease, myasthenia gravis, and dermatological conditions, and transplantation, rheumatic diseases, neurological diseases, many neurological diseases, including myasthenia gravis, CIDP, or there's a disease called multiple sclerosis, which is a very dangerous disease, and in conditions like autism, schizophrenia, Parkinson's. How? That means there was no logic. You started using it. And then one day I have been in the flow of these studies. And one day I was in a meeting in Washington, and there was a group of people, doctors, who were using IVIG. This is called IVIG now, okay, immunoglobulins, right? So we were using, and then what are the new uses? One doctor says, from Gelfand, he says, we can use IVIG in chronic fatigue syndrome. And I asked, Arvind, why would you want to use IVIG? What's the rationale for using immunoglobulins in chronic fatigue syndrome? He says, why not? Why not? I mean, American accent, okay? Why not is not scientific. You need rationale, you need meaning, you need to know how it works. And it is, if you start using why not, then you will have an enormous requirement. There will be scarcity. People who really need it, they will not get it. Because you know how many Alzheimer's patients are around? How many elderly people are there? Look at us, look at us. Yeah. So we all need these antibodies. If you start giving antibodies, these old useless people, like me, what about young people who really need primary immunodeficiency people? You will not be able to. So this is why we need to understand how these immunoglobulins work. What my lab has done all my life, I have devoted my life to understand how these very normal immunoglobulins in you all, we all, how they exert this beautiful mechanism of helping in autoimmune diseases. And autoimmune diseases are very complicated, and the mechanisms are equally complicated. Today I will not test your patience. I will come back. I'm sure Professor Hanumantajar Joshi will invite me. I will explain to you how these immunoglobulins work. But a very quick glance, okay? So they work through the many mechanisms, many routes, many pathways. These are all immunological pathways. I will make it easy and short, okay? There are hundreds of papers that we have published, and what I would like to insist is the fact that the cellular and molecular mechanisms of how IVIG works is interesting, there's no doubt about it. But for that, we need to sit down a little bit closer with more attention, with a little bit more background in immunology, the role of different cells, the role of different molecules. We will be able to appreciate better the mechanism of action of IVIG. We have made some inroads, but what I would like to stop, where I would like to stop is, although these beautiful immunoglobulin molecules, they're used in many diseases with very beneficial effects, they're expensive. They're very expensive. For pharmacology people, you may be knowing IVIG, one treatment of 4 or 5 days is, it costs roughly 2 lakhs or something like that. You should be knowing, doctor. Huh? You use in rheumatic diseases. We'll talk about the mechanisms in detail. So it's very expensive, but it would be good to know whether we can give it to this patient or that patient. That means it makes more sense rather than randomly giving such expensive drug, not just because it is expensive, that is a real problem, but also the body has to take this huge amount of antibodies. It's a burden, and we don't even know whether it works or not. In 70% of the patients who suffer from chronic inflammatory demyelinating polyneuropathy, it's a neurological disease. It works. 70% of the patients, they recover quickly. But 25 to 30 patients, 30% of the patients, they do not. But we don't know in the beginning. If you knew who is benefiting, whom we can give or not, are you with me? If you know in the beginning itself, okay, you can give it to this patient, he will be beneficial, don't give it to this patient, the person will not respond, this patient will not respond, it would be good to know. But how to know? How to know? How do you know beforehand, before even you give? And this is where I'm sure in your college also you are learning artificial intelligence, machine learning, deep learning. These are the novel or modern approaches of understanding the signatures of each individual. We all have a signature of our immune system, and any change in our immune system, our immune behavior is reflected in these signatures, and that is the beauty of artificial intelligence. You can use artificial intelligence to make myself more handsome looking or like Shah Rukh Khan, but that's not much useful. I can also have some hair here on my Photoshop picture. That's a different story. But imagine the beauty of artificial intelligence that assesses thousands of people like us. You have the data from thousands and thousands of healthy people, people with diseases. What is the difference in the signature? And those that responded, those that did not respond, is there a meter, a parameter, a biomarker that can be different?
So it means to say without AI, you cannot identify as it is.
Very good question. You can, but the number of the patients that you would require to have a statistical strength would be very difficult. Many people have tried in myasthenia gravis, in Guillain-Barré syndrome, many diseases IVIG is used. What they do is, I have done this study. We take the blood and we do the transcriptomics, genomics. There are quite a few omics that you can do now. And you see in 10-12 patients, but there are 10-12 markers that are differing. And there are some overlapping with the healthy people, some are overlapping with the patient. You're not getting a clear picture because the strength of statistics is not good enough. When you look only at 5-10 people, you are all pharmacy people. Whenever you try to discover a new drug, you cannot give it to 4 or 5 people and say it works. You need a very strong statistical strength. That is the reason why we need to do clinical trials, placebo, blind, double-blinded, control, everything is there. Only then you can say this molecule is powerful and it's going to work. And until then, it will be just an observation. And even then, even when you do all this, all the patients do not necessarily respond to all the drugs the same way. We all human beings are very different. There is polymorphism. We have a genetic diversity. We differ and our responses will be different. And therefore, the larger the cohort, larger the statistical strength, more powerful, more reliable will be. That is the beauty of artificial intelligence, because you just subject the data, huge amount of data, it will do churning like that and it gives you the substance, it gives you the key. We hope so. I mean, I'm not saying artificial intelligence will do everything, data learning, machine learning, etc. But some deep learning is going to be useful. And I hope that we'll find that one day. So I'm going to stop this by giving you 2 messages. First message, I hope you understood that we all have antibodies, and we all have these antibodies that fight against the infection. As you're sitting here, they're working. So the antibodies are a must to fight the infections that you encounter every day. Okay? And this is a knowledge that you already probably knew. But the second message that I'm trying to pass on to you today is that very same normal immunoglobulins or natural antibodies that are you and me, we have, they also have very strong immunoregulatory
immunomodulatory functions in autoimmune and inflammatory diseases. This is original. This is the message that I want to pass on to you. Meaning that you have antibodies, you have 10 milligram of antibody right now in your body. It is not fighting only against infection. Okay, it is doing something else. And that something else is still mysterious. We know that these antibodies are powerful. They are having immunoregulatory functions. Okay, but this we have not fully understood. We will have to work more on that. And hopefully we will. So to end, what I would like to show is this. This picture there is a simple picture where you have like a Kukkarhalli Kere. One particular moment I took a picture on a cool, normal, simple, no stormy day. If I take a picture of Kukkarhalli Kere, this is how it is. For me, the normal Kukkarhalli Kere, the ideal day in Mysore, like most days in Mysore are ideal days. It's a beautiful city, right? Your immune system on a perfectly natural, normal day looks like that. Your immune system is not confronted with a pathogen, confronted with an aggression, confronted with any challenge. It looks like a Kukkarhalli Kere. But the day you put a big rock onto your Kukkarhalli Kere or a little tremor, then it gets dispersed. In the same way, your immune system, whether it is a single clone or a small molecule that changes, it soon becomes highly rampant. It starts, you think it starts, autoimmunity starts with one antibody, one autoantibody. No. Soon, the entire dynamics of the immune system changes. The very architecture of the immune system changes, and that can be measured by mathematical computations using Fourier's transformations. These are very useful, strong tools. You must have read when you were young. Fourier is French, by the way. Joseph Fourier, Frenchman. Just a little French. So, what I'm trying to tell you today is that you all have these natural antibodies that are functional right now. They all have a job of maintaining homeostasis. You know what is wired? W-I-E-R.